Good day. Just a correction. Can everyone? Hope everyone can hear me. Just a slight correction. I am essentially retired from the Research Foundation. I am the Canadian liaison for our subscribers in Canada, and I'm doing this as a pro bono for our good friends in Canada. I did work for the foundation for 25 years and retired for three years ago, and have done this presentation many times, and in fact I've made 80 or so trips to Canada in the last 20 years, and will be doing 5 or 6 more trips this year. And I want to thank the foundation here in Denver for allowing me to use their facilities here, because they have a much better, faster network than I have at home. My presentation today is based upon the published science, and may be contrary to some of the things being taught or understood as embodied in the current regulations or guidelines. And often the regulations, both in the U.S. and Canada, may not reflect the science, and that's the case in many instances. And after 40 years of experience in drinking water microbiology and witnessing the politics in the formulation of drinking water guidelines or regulations, both in Canada, U.S., and worldwide, I believe some of my recommendations, even though they're contrary to the regulations, still have a legitimate basis. So I'm the messenger of the science, not necessarily saying that the regulations or guidelines are always current or accurate. So with that, let's launch into the presentation. As Nicole had said, I will be happy to take questions throughout the presentation. I believe we have about an hour. The presentation is in 3 parts. First of all, I want to go through what I call microbial myths that have been perpetuated for decades in the last century and seem to be being propagated in this century. I want to go through the indicator concepts and why we have it. And then I want to go through recommendations for public health protection, because that's our goal as public health protectors, is how to protect public health in drinking water. Again, I have this nice little cartoon that I have borrowed called The Attack of the Myth Makers. The myths. I'm sure there may be other ones, but these are Marty's favorites. First of all, that pathogen monitoring ensures public health protection. The second, all bacterial indicators can be used to make equivalent public health decisions. Third, bacterial numbers in drinking water are relevant for public health decisions. And I'll go through these and explain them in further detail. Again, my presentation, I have cartoons that I think help liven up the discussion and hopefully point to the point I'm trying to make. First of all, we have early microbiologists looking for pathogens. And now we have modern microbiologists looking for pathogens. And if I could use this pointer here, I want to show here that these two the previous slide showed the early microbiologists. The only difference here is fancier equipment, but I want to point out the trial data and the error data, that the error data is quite significant. So again, the difference between modern and early microbiologists is more fancy equipment, but the data is about the same quality. Now recall the purposes of water treatment, and I've summarized them, and maybe the list could be a lot longer, but I try to make this very simple because of our time constraints. First of all is to remove or inactivate all human pathogens, that is protozoa, bacteria, viruses. It's not to produce sterile water. So water that comes from a treatment plant is not sterile. It contains other organisms that are not pathogens. They're just normally occurring bacteria which have no health basis at all. But we want to remove the pathogens. We also want to remove suspended particles, reduce organics that can form DBPs, reduce taste and odor, reduce color, etc. We want to produce water that is aesthetically pleasing, safe to drink, so the consumer will drink the water and not necessarily run out to buy bottled water, which people will do anyway, but that's the purposes of drinking of water treatment. Now the barriers for pathogen monitoring are numerous. There are technical issues, administrative issues, demographics, the quality of data, and the representatives of data, and I'll go into these in a little more detail. But there are barriers of pathogen monitoring, and you probably know most of these, but I want to at least refresh your mind on what these are. First of all, the technical issues. Very low numbers of pathogens in source waters, very low numbers in wastewaters compared to the other numbers. There's billions and trillions of bacteria, of organisms in source water, and there's very low numbers of pathogens. They're very difficult to detect. Pathogens is very difficult to determine viability, whether they are infectious or not. They may be present in source waters, but are they viable? Whether they are alive, whether they are infectious, whether they're human, whether they're non-human. We also need to sample very large volumes of water in the order of 10 liters or more to have any hope of finding pathogens, protozoans, bacterial or viral, in these waters. There are also very few labs equipped or staffed to look for these organisms. And it takes days to weeks to get the results. Not necessarily these results are valid, accurate, or reliable, but to get a number. Here's a table from a paper I published in 2002. The name of the paper was The Plain Hard Truths of Pathogen Monitoring, published in the Journal of AWWA, which won the Best Paper of the Year award. It's a list of treatment plants in Milwaukee, Ottawa, Carleton, Calgary, Denver, Epcor, which is in Edmonton, Windsor, Fort Collins, Colorado. Again, looking at how much water they produced, how much water they sampled, the number of samples per month, total of water sampled per month, and the percentage of water they sampled compared to the water they produced. A pretty small amount of water they sampled compared to the water they produced, using very, 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 very poor methods. And we're supposed to do a leap of faith saying, based upon the amount of water they tested, using very poor methods, they're supposed to say the water is okay or not okay. Doesn't make sense to Marty. Maybe it does to someone else, but not for me. Okay, what makes microbial data credible? These are the three criteria. Sensitivity, a true positive. This says that if a pathogen, using a method, if a pathogen is there, you'll get a true positive. Specificity says if a method shows that it's a negative, it's a true negative, there is no pathogen there. And reproducibility says if I use a method and I use it in, say, Halifax, and I use a method, say, in Victoria, or I use a method in Calgary, whatever, if I use the same method, I'll get comparable results. Those are the sort of the gold standards for a method. And there are no methods for pathogens that have these three criteria to make timely public health decisions. Now, the EPA and all their wisdom, I worked for them for 11 years, thinks there are methods out there, but frankly they're not. Okay. Since, in my estimation, there are no good methods for measuring pathogens, and there are dozens of pathogens, and so which ones do you want to pick to make public health decisions? Since there's protozoan, bacteria, and viruses, so we can't measure for all of them. So we went to the idea of we can't measure for pathogens, we use bacterial indicators to say whether there's a probability of pathogens being there or not there. We made this idea with microbiologists back in the 1800s says, let's look at indicators. Maybe that's a better idea. Surrogate. Indicator concept. I'm sure you heard this before, but I'll go through it very quickly. Since it's impossible to monitor pathogens, all pathogens, let's use an indicator that signals recent fecal contamination, that is increased fecal load in watershed, water treatment failure, contamination during storage or distribution. Indicator organisms always present in fecal material. That's good. That's a good idea. Indicator organisms persistent similar to pathogens, that is days to weeks. And detection methods relatively simple, inexpensive, and rapid. Because we can't wait days, weeks, and months to get results to say this water should not be consumed because it may have pathogens in it. So these are sort of the basis for selecting an indicator which would give us some idea as whether the water should be consumed or not. So we have three indicators that we've used through using now and using in the last 2 centuries. Total coliforms, few coliforms, although the correct term is thermal tolerant coliforms, and I'll get onto that later on, and then Escherichia coli. Those are the 3 common indicators that we use or have used in the last 2 centuries. Fecal coliforms, and there's 2 spellings, and I've used the U.S. spelling. It's theorized that 44.5 Celsius rather than 35 Celsius would screen out the non-fecal coliforms. Good idea, doesn't work. And the reason it doesn't work is in subsequent research that the Water Research Foundation and others have shown that Klebsiella, which is a coliform, folks, at 44.5 degrees grows Klebsiella, 15%, thereabouts, which is of not a fecal origin. So we have a 15% error here. Klebsiella likes to grow in forests and likes to grow in prairie environment. So we have 15% error rate from Klebsiella growing at 44.5. Also, we found that, and this is some of the foundation research, that 10% of E. coli does not grow at 44.5. E. coli, of fecal origin, doesn't grow at 44.5. So we have a potential error rate of 30% using fecal coliforms as a surrogate. So we have a 30% error rate. Isn't that great? So we probably should not be using this as a indicator of safety of drinking water because we have a tremendous error bar. Some people still think this is great, but I would recommend, you'll see later on why we should not be using this either in drinking water or in source water. All right, here's some take-home messages if you're taking notes. Coliform genera, there are 5. Escherichia, and I have it in red because it's important being read. Human and animal feces, 94+% of bacteria and fecal material are Escherichia, okay, 94%. Enterobacter, environmental and fecal, Klebsiella, Citrobacter, and Serratia or Serratia. Those are the 5 genera that comprise coliforms, right? And I have some data later on that'll help convince you, I hope, of why we look for Escherichia as opposed to the other four, which are sort of just tag-alongs. All right, I hope you can read this table here, but I'll go through it anyway. Okay, this is a table that shows, and I'll use my wand here, all right, human feces, animal feces, water, milk, grain, and soil. Number of observations. This is in 1921, okay? That's before I was a young kid at that time, 1921. Looking at, this is the Aerobacter, and we renamed it again. Us microbiologists do this to confuse the engineers. It's Enterobacter now, but Enterobacter orogenes, and you see there's 5.9% Enterobacter in human and animal feces, and down you look at soils and grains is up to 88%. Where we look at human and animal and human feces, we look at 94%, 92%, whereas E. coli in grain and soils is down to 18, 11%. Okay, so remember, it's 1921, and we're looking at 94%, 92%. Okay, so it's 94%. Do you remember that, to ladies and gentlemen, to the next slide? Okay, that's 1921. Whoop, let me go back here. Okay, 1977. Al Dufour, a friend of mine from EPA, looked at a number of animals, looking for E. coli, Klebsiella, Enterobacter, and Citrobacter. He looked at a lot of animals here, domesticated, human, horses, averaged 94%, 94%. E. coli. Klebsiella, not much, folks. Enterobacter, Citrobacter, not much. So the magic number here is 94%. Again, 1921 and 1977, 94%. So that tells Marty, who's not a rocket scientist, that 94% of organisms in fecal material, human and animal, is E. coli. So that's probably the one we should be looking for if we're looking for pathogens that are associated with fecal material. Again, E. coli always hangs around with pathogens. This paper from 1885 by Theodore Escherichia on Bacillus coli. Remember I told you that us microbiologists keep on changing names? Bacillus coli was renamed E. coli in honor of Theodore Escherichia, who said that Bacillus coli was the best indicator of pathogens in drinking water. 1885. But we had no methods at that time to speciate specifically for Escherichia coli. That wasn't until the 1980s when the Water Research Foundation was able to develop a test for Escherichia coli that was cheap, simple, and rapid. So we had the science in 1885 pointing to E. coli, but we didn't have the methods to do it. So that's why we had to go to other indicators, and that's why we went to total coliform, and that's why we went to fecal coliform, because we didn't have the tools to measure E. coli. So, why do you measure for E. coli? It's always present in human animal feces from those earlier tables, 10 to 8, 10 to 9 per gram, correlates with fecal material, persistence, similar to pathogens. Again, I mentioned the rapid methods now we have, inexpensive, easy to perform, even for the small utilities. So now we have the tools to measure for this critter in 18 to 24 hours. All right, regrowth here. And this ties in what the flower was on boil advisories, why we have them so often, and why they're put on and why they're taken off. From the earlier slide, you saw that Klebsiella and Enterobacter were coliforms. When water is treated, there's always some organics that are present in the water, and these coliforms colonize within the water systems, the pipes, and the storage tanks. It's a common phenomenon, and they're always in there, and they attach and they colonize. And the coliforms that do this most often are Enterobacter and Klebsiella. And they're promoted by nutrients and warmer temperatures and low flow and low distribution, low disinfection residuals. And there's a lot of research the foundation has done on this. It just shows that when you have organics in the water and you have warmer temperatures above, say, 19 Celsius in the summertime, you have low dead-end mains, you have Enterobacter and Klebsiella growing, and they slough off. And if you're unlucky, you take a water sample and you get coliforms, and you think there is a potential health hazard. What's the first thing you do? You maybe issue a boil order. And often your public health officer may be not knowledgeable of the difference between E. coli and coliforms or fecal coliforms. He or she may think this is a breach of health, increased health risk, and issue a boil order. Or in fact, you just have these Enterobacter and Klebsiella growing in the system, and you go to a boil advisory, and you do resampling, and all kinds of things happen, and really there's not a health problem. It's just you have coliforms in your system. And unlike fine wine, water does not improve with age. As water leaves the plant or goes to the system, the coliforms grow. You have higher turbidity, disinfection residual goes down to maybe 0, and so it deteriorates with time. Here's an electron micrograph. I hope you can see it of something I did back when I was at EPA in the 70s, water main tubercles, and in there you can see these are water. These are tubercles. These are nodules that grow in the water main, and these are bacteria which are growing. They may or may not be coliforms, but they're happy bacteria growing. They love it there. They found their spots. Heard of a place that they set up shop, and they just grow in there. And if you have a hydraulic shock or a water main break, you get some of these things sloughing off. And if you pick them up in a water sample, they may be coliforms, and you get a positive sample. Here's a larger magnification. This is New Haven, Connecticut, and there's some flagella there. So anyway, this is what happens in all the water systems throughout the U.S., Canada, and the world. Again, this is Klebsiella. He lives in distribution systems. He has no health significance. There has never been any health significance attributed to Klebsiella. In fact, one of your fellow Canadians, I.B.R. Duncan, University of Toronto, looked at Klebsiella in drinking water. There's no waterborne disease ever associated with Klebsiella in drinking water. They're all been hospital-acquired nosocomial infections. So Klebsiella has no relevance as far as health effects or health risk in drinking water, period. And so it's a coliform. It grows in water systems, and therefore it should be discounted as having no health significance, period. Regrowth, ramification, more corrosion, loss of disinfection residuals, increased turbidity, taste and odor complaints. I spelled odor wrong, I'm sorry. It should be O-D-O-U-R, I'm sorry. Increased frequency of coliform positive samples, and unwarranted boil advisories. So I just hope you take that to heart, that you have to be careful on the boil advisory, because it's really economic hardship and a lifestyle hardship for your communities when you have to go to a boil advisory because you have a coliform positive sample without the repeated presence of E. coli. Ramifications of public health advisories, and this is what we had put in the when I was with did a report with the technical advisory group for the Minister of Health in British Columbia a couple of years ago. Economic hardship: schools, hospitals, dental offices, restaurants, tourist hotels. Scalding. One of the things you should consider, if you have a hot water heater and it's at least turned up to 65 Celsius, hot water heaters are essentially pasteurizers, so they pasteurize and you have, when you have hot water at 65 Celsius, has essentially killed everything at the sterilizer. Again, the problem is there's no definitive endpoint when to end advisories, often lacking. It's like, yeah, well, after 2 weeks it's probably okay and call it off. So it's really difficult. It's easy to put one on. It's just difficult to say exactly when to turn it off on an advisory. So be careful. Again, Enterobacter, a good buddy of Kleb, lives in the system, no public health significance, in place poker. Okay, this is a question that I have for you just to think about. So why would one use total coliform as a public health indicator in drinking water, knowing that this group of organisms found in the environment free of fecal contamination, and these organisms colonize and grow within the drinking water distribution systems? And since coliforms grow in water distribution and storage tanks, why would numbers be relevant? And this is what I keep on asking people. Okay, enumeration methods. If you really think or have been told that numbers are necessary to make correct public health decisions, wrong. When I was with EPA, I worked with a gentleman called Edwin Gelreich, and he's the gentleman back in the 50s and 60s developed the membrane filtration method. And at that point, there was this consensus that numbers, the number of coliforms on a membrane or the number of fecal coliforms on a membrane, was tied directly to risk. The number of bugs on a membrane was significant. The more membrane bugs, the higher the risk. Subsequently, back in the 60s and 70s and later on, it was found out that this assumption was not correct, and that enumeration methods were false, because the bugs in the water are not uniformly distributed. Membrane filtration always underestimated the numbers, and actually membranes were defective, that they underestimated the numbers, and that heterotrophic plate count bacteria greater than 100 CFUs per milliliter, that's my research, interfered with enumeration methods. And there was a wide variety of microbial data showing that enumeration methods for the use of determining potential risk was on a slippery slope and could not be justified. And back in the 80s, mid-80s, the US EPA went from a number concept philosophy to a presence-absence concept. And one of the driving forces was research done by Wesley Pipes, in which he took a well-distributed water sample and took samples, and it's hard to read here, but what Dr. Pipes found out that taking water samples either in a laboratory or in your distribution system is more like a crapshoot. You have one chance, you have a 37% chance of getting one bacteria, 37% chance of getting no bacteria, 18% chance of getting two, and 7% chance of getting more than two bacteria. So it was more like a casino, you know, it was luck of the draw. So based upon the casino environment, EPA went from, rather than having one bacteria or two bacteria or whatever, determining whether water is safe, less safe, or not safe at all, to a presence-absent concept. That is, if you have one E. coli, it's a higher risk than none at all, and whether you have 10 E. coli or 9 E. coli, it's the same risk. So that's why they went to presence-absence. And then just to step back a moment here, one of the things that I've always been picking on with microbiologists is that when they use membrane filtration and they go and they have 3 or 4 colonies on a membrane, often they don't go through the completed 72-hour completed tests for presumptive confirmed completed. If they have a positive colony on a membrane or 2 colonies on a membrane, they don't follow the completed 72-hour test. And also they don't use the confidence interval, which on the next slide, that shows what the wide variety is of the wide variety of confidence in a number. This is one of the reasons, and I'll go in the next slide, it shows you why Health Canada took out the number in their guidelines. If you look at standard methods, and this is one of the driving reasons why numbers are really not very useful at all. Earlier guidelines in Health Canada showed that if you had less than 10 CFUs per 100 mils, you were okay, but 10 was not good. Well, the confidence interval for 10 is 4.7 to 18.4. That's a huge range. So 11 was bad, but 9 was okay in the old Health Canada guidelines. Well, actually, they're the same number. And EPA also looked at this wide range and said, this is a bunch of smoke and mirrors, and this is just not relevant. That's why one of the reasons they went to you have less than 1 or 0 is okay, and more than 1 is the same as 1, 2, 5, 6, or 8. And this is why my comment here is I told why Health Canada dropped the 10 CFUs per 100 mils from their guidelines. HPC interferences. One of the other reasons why membrane filtration or enumeration methods, you use enumeration methods to get a number because you think numbers are important, but HPC, heterotropic plate count bacteria, one of the problems with HPC bacteria is that they reduce the accuracy of lactose-based membrane methods. And if you have high HPCs, you can have a 0 on your membrane, but not because there's no E. coli or there's no coliforms there. It's because the HPC, if there's high numbers, have actually masked, they've interfered with the ability of the coliform or the E. coli to grow, because there's just so many HP bacteria there that they just mask the ability of that organism to grow. So if you're going to be using lactose-based membrane analysis, you do parallel HPC to make sure you don't have high HPCs that are actually giving you false negatives. So, my final conclusion, bacterial numbers have no public health significance in drinking water. If you're looking for E. coli and you have 1, 2, or 3, it's the same thing as having 1, which is not a good sign, and having none is the best answer. Recommendations, and I'll go through these, and again, as I mentioned earlier in the start of my presentation, my recommendations may not be consistent with the province or Health Canada or with the state, U.S. state or U.S. EPA. I'm just saying here are my recommendations, and based upon the science and my 35 years of observations, and my point is how to best protect public health. Okay, so I'm just saying here's where I think we should be thinking of how to protect public health in the best way possible. Again, this is what I think where we should be doing to protect public health. And these are the things I think should drive public health decisions. One of the first things I've been struggling with, both in the U.S. and in Canada, I really believe that with the new tests, and I'll go through these later on, is that the modest-sized public health systems who are trained to operate their plants, take water samples, can reliably do their own compliance monitoring tests. They take pH, they take turbidity, they take disinfectant residuals. They are, to me, capable. Not the smallest ones, but those who are trained are capable of providing an enhanced level of public health protection if they can do their own compliance monitoring on site themselves. And in a paper that I hope to have published this June or July in the Journal of the American Water Association, in Alaska, in Ontario, in Quebec province, up in the First Nations, they allow trained operators to do this. Use the Coliform group to monitor quantum quality changes, not health changes, but are there high quality, are there increased organics in the water? Are there breaches of the integrity of the distribution systems? You can use the Coliform group as a water quality surrogate. So the Coliform group can be used for those purposes. There's a new group of methods called the chromogenic enzyme methods that monitor both coliforms and E. coli simultaneously. These give results in 18 to 24 hours. We should be embracing those. Use HPC to monitor for water quality changes in the distribution system, looking for quantum changes. We're going to say you have 100 HPC, typically maybe you're looking at tens of thousands. That tells me there's something happening in your system or your source water. So we can use HPC monitoring to measure changes that are not necessarily health related, but maybe organics or different changes in source water quality. Use lipid-based methods. As I mentioned, membrane methods are susceptible to clogging and colony spreading. Liquid-based methods always, or almost always, are more sensitive. They pick up higher colony counts, so they're better sensitive to picking up E. coli or coliforms, so they're better than membrane methods. Understanding that all micro methods have some variability. So you need to avoid public health decisions based upon a single sample. So if you find an E. coli positive sample, I would avoid going to public notification or board advisories based on one, because because you may have an erroneous sample. But I would certainly use E. coli as the best surrogate, but not necessarily go to a board advisory just on one sample. After a second sample, I would consider a board advisory, but again, look at the sanitary survey of the system, other history of that system. Again, board advisories are very, very serious, so be cautious about just saying, let's do it to be safe. I think that's what happened post-Walkerton. Everyone went to a board advisory just to be safe, but it sure is a hardship on your community. Use HPC and disinfection visual data to identify areas in your system that may have long residence times and thus allowing for regrowth. Strive to eliminate dead ends and sections of your distribution system if that's possible. Another thing that's happening more often in the States, maybe in Canada, choose your sampling sites very carefully. Sometimes you pick a sampling site like Luigi's Pizza, and you get a positive sample not because the water is bad, because the sampling site was bad. Maybe you should look at having dedicated sampling taps at a fire station or a police station or the city hall, but have that in a lockbox. Have that water running all the time before you take the sample, not using a flame, but actually using an alcohol wipe at the spigot. What you want to test is the water that's in the system, not the water that may be contaminated at the sampling port, because if you get a bad sample because of the sampling port, then you have to resample. If you get a positive sample, then you have to do all kinds of resampling, and you get unwarranted public notices. So that's there's a lot of things riding on a bad positive sample that's not related to the quality of the water, but because of the bad sample taken by the sampler or the sample contamination caused at the sampling port. Please discontinue all fecal coliforms to quantify fecal loading in source water. Use only E. coli. Discontinue use of fecal coliforms for drinking water, please. And use E. coli methods don't require confirmation steps, days to complete. These methods just are so old school and so bad. They give you erroneous results, take too long, and they're just such good tests out there that you can use right now that are cheaper and better. Again, use simple methods to provide credible data to make correct public health decisions in the shortest possible time. Use operators. Operators should be trained to perform microbial compliance monitoring using approved CE methods. Okay, here's some considerations for using these CE methods. There are 5 of those that are approved or accepted either by the US EPA or approved by standard methods. Just for background, 75% of Canadian and 93% of US public water systems serve less than 10,000 population. Operators are already certified to measure water quality parameters. They're your first line of defense. They're there all the time operating these plants. And they can sample every day and don't have to ship samples by courier in the dead of winter. These methods are robust, simple, inexpensive, no courier costs, and they provide public health protection data in less than 24 hours. They're used in Quebec, Northwest Territories, Alaska, and approved communities. They're already used in certain parts of Canada, in the U.S., and Alaska. Here are the list, and I'm not selling any one of these. I'm just saying these are out there: CoLert, CoLassure, ReadyCult, E. coli test, ColiTag. These are the earlier ones. These are for coliforms. If you want to use coliforms, here are the ones that are for E. coli. Here's CoLert. This is an example, and CoLert was developed by the Foundation back in 1898. Just an example. The bottle in the back is clear, which means there's no coliforms and no E. coli. This sample here was positive for coliforms, and under fluorescent light, it's positive for E. coli. So it's a 100 ml sample, incubated at 35 Celsius. If it's yellow, it's coliforms. If it's fluorescent, if you take this and put under UV light, if it's fluorescent, it's E. coli. Equipment required is, again, air incubator. Again, often local clinics have an incubator and can accommodate the needs of the water utility. A UV lamp, media packets are less than $5, and disposable sample bottles. Pretty slick. If you want more information, more than I've given you here, I wrote a paper for a symposium this past April. And after this webinar is done, I'm told that Nicole that this is available. You'll have these slides. The Journey of Microbial Monitoring for Ensuring Safe Drinking Water. This is about a 30-page document that goes through more than I've given you here, and much more references if you wish to have that. One of my favorite slides. By the way, you guys did a great job on the Olympics. I'm glad you guys won the gold medals in hockey, although I hear the women hockey team also got a gold medal in beer drinking. Good for you guys. And that's all. So we got 15 minutes for questions, and I'm going to turn it over to Nicole.
Okay, so we have a question from Albert Simon. So go ahead, Albert. So Albert Simon from the Ontario Ministry of the Environment would like to ask, do you see a use for coliphage in groundwater?
Good question. I guess I don't see the use for that. I would look for I would look for more. I would rather look for cut to the chase and look for whether E. coli is there, because that would indicate to me the presence of recent fecal contamination. Coliphage, since it's a virus, could be there for weeks, months, or years, since it could have been contamination that happened in years past and could be dormant because groundwater is a protective environment. It's cold. It's no predators. It doesn't tell you when that coliphage could have been when it first contaminated the groundwater. Whereas if you had E. coli in there, that would tell me that there has been contamination coming either through a faulty seal along the casing of the well, or from infiltration above from surface water.
Okay, Huron County had a comment. They said that it's very informative and a little scary. They issue a lot of boil water advisories based on total coliform data. Albert Simon from the Ontario Ministry of the Environment has another question. What about secure water that is intermittently contaminated with virus-sized particles?
I guess I'm not. Can you clarify, Albert? Could you clarify that question more? I'm not quite sure what you're asking.
He says he's still talking about groundwater.
I'm still puzzled as to what exactly you're asking me. Maybe you could rephrase your question. While he's doing that, I guess I'm still concerned about why one would issue a boil advisory based upon coliform. Hopefully, now knowing that coliforms include Klebsiella and Enterobacter, which inherently grow in distribution systems, and these two genera are environmental strains and have no relationship at all to public health, that may no longer happen again.
Albert says the absence of E. coli in groundwater does not rule out the presence of viruses.
He's correct. The other point is that what I would be assuming is that systems that use groundwater also have at least some type of disinfection. In the U.S. under the groundwater rule, public water systems that use groundwater have at least disinfection, and disinfection does take care of viruses.
Albert Simon now asks, how do you feel about Enterococcus? Sorry, I'm not sure if I'm pronouncing that correctly.
Enterococcus is another indicator that has been used on occasion as an indicator of fecal contamination. It has been, the consensus has been that E. coli is a better indicator of fecal contamination, and therefore is the preferred indicator for drinking water. Enterococcus has been used more often or has been used also for bathing beaches more often than E. coli. So the Enterococcus has been used for bathing beaches, salt water more than E. coli, but again, E. coli has been selected, or the consensus has been E. coli is the best indicator for freshwater drinking water.
And there's a question from Stephanie Gerard from Health Canada. Go ahead, Stephanie. Oh, I see. Any new information for developing surrogates for Giardia crypto?
I'm not sure how long we want to keep on chasing your tail on this. As far as surrogates, I guess in 1992, I don't want to just always focus on EPA, but they seem to spend a lot of money on things that may help help Canadians not waste their money. Excuse me. Let me rephrase that. In 1992, the EPA required the U.S. utility to spend, I think, $125 million called the Information Collection Rule, in which they required the utilities to look at their source waters for Giardia and crypto. They did 5,800 samples using crappy methods, terrible methods at that time, and found 83% of the water samples did not contain Giardia or crypto. These are source waters, and we know for a fact that all source waters have crypto or Giardia at one time or the other. So the methods were very poor. But in the data that we analyzed, there was indication that E. coli was probably a good surrogate indicator for crypto or Giardia, and there is in the works I hear that it may be that E. coli is still going to be a good indicator of the presence of crypto and Giardia. That said, it's saying that the more E. coli you have, probably the greater probability of having Giardia and crypto. That being said, that rather than looking for more surrogates, it's probably important to say that if you have adequate treatment for source waters, and adequate treatment is filtration, membranes, UV, ozone, chemical disinfection, that series of toolbox of treatment to achieve the log reduction you need to remove Giardia and crypto, you've done your job. And using adequate treatment to achieve the log removal, to remove what you feel is the worst case in your system, you're going to be okay. And I hope I didn't touch something that I wasn't supposed to touch on my system here. Okay. I hope that answers your question, but again, E. coli appears to be the best appears to be at this point a reasonable indicator of whether crypto or Giardia will be present.
Dr. Allen, several people are asking whether the PowerPoint will be made available by, if I can email it to them after the presentation. Is that possible?
What I was wondering whether I could do this or not. This is a sensitive area here. What I would like to do, Nicole, is I don't mind email presentation. I would like to sanitize it. Some of the cartoons that I have in here were given to me by the cartoonists in that. So if I could, Nicole, is after this is send you a revised revision, and then you can send these out to the attendees. If that's agreeable, I could be happy to do that.
Okay, so for those of you who want the PowerPoint, that's what's going to happen. Dr. Allen is going to remove some of the slides that have cartoons on them because of copyright laws and things like that. He will then send it to me, and I will then send it on to you. So there's 3 people who have requested that so far, and if you want a copy, you can either email me, it's info@safewater.org, or let me know right now. Stephanie from Health Canada says that she strongly disagrees that E. coli is a useful surrogate for crypto and source water. She's basing this on recent literature.
Well, Stephanie, I don't know what you want to use for a surrogate. Do you have some suggestions besides E. coli?
Okay, I'm sorry, I'm confused, Stephanie. You say, no, not you, the other one. Oh, sorry. Yes, there's 2 Stephanies. So not Stephanie from Health Canada, Stephanie, not from Health, also from Health Canada. So no wonder I'm confused. So Dr. Allen's wondering what you would want to use as a surrogate then. Okay, so Stephanie says that she's with the Bureau that develops the water guidelines. Stephanie, the question still remains, what should be used as a surrogate then? Okay, so she's hoping that you, Dr. Allen, can tell them what should be used as a surrogate. Albert believes that E. coli is used as a surrogate in small systems in the U.S. and New Zealand.
You know, we can go round and round and round on saying, use a surrogate, use a surrogate. I'm more of a practitioner. I'm saying that we know what treatment processes are able to remove what level of contaminants. And rather than chasing our tail of what surrogate is best and what surrogate does what, I'm of the thought processes of let's just get on with it. We have all these treatment tools to remove contaminants to various levels, and just get on with it. Again, as I mentioned before, we have processes now that are very affordable, even for the smaller systems. We have UV. We have package plants for membranes. We have small ozone plants. We have different types of disinfectants. We have the MIOX processes. And rather than waltzing around what surrogate is best, what surrogate is the better ones, I just can't understand why we just don't stop fishing and cut, stop cutting bait and fish and just do it. And E. coli is just one way of measuring fecal loading, fecal contamination. If I have 10 of the 6, 10 of the 7, 10 of the 8 E. coli per 100 mils, I know I have a major fecal loading problem in my source water, and therefore I have to have pretty robust treatment processes to remove this. If I have pretty pristine waters, which may I find up in British Columbia, with turbidity excursions of maybe 1.5, 2 turbidity units in the spring, I don't have to have as much filtration. I can get by with maybe UV or less exotic treatment processes. So the engineering and the science is there. I just don't understand why we just don't roll up our sleeves and just do it. I guess he's frustrated with having to keep on looking for surrogates when E. coli seems to be the pretty good workhorse, and just use it. I'm sorry if it offends some people if we have to do more science to look for more surrogates when I think we really have one that was recommended back in 1885.
Albert is asking what level of removal in activation is good enough. You need to characterize your source water.
And we have, by characterizing your source water, are you suggesting you know what the level of contamination is? That is, are you finding what is the level of waste of fecal contamination, or what's the source of contamination?
He says for Cryptogiardia.
Is this for a utility looking for what type of treatment plant they need to install?
Albert says that they have minimum requirements, but how would a water treatment plant know the level of removal that is required if it doesn't test for specific pathogens like crypto?
I guess to answer your question, and I'm not sure how to best answer this, is that there should be just a default decision saying that if we install these type of processes, we know we're going to get 6.5 or 7.5 or whatever log removal, and do it as just a worst case decision here without having to go through all these pilot testing, without having to look for crypto. At some point we have to stop. We have to say this is the worst case scenario. These processes we know will remove these organisms to this level, and just do it rather than having to trying to do pilot testing, do analysis. And again, if this is done in a way in which then all water systems using this source water use this default, rather than having every system have to do their own worst case scenario, I think you can move on to just building the plants without having to provide this piloting and analysis to satisfy every regulator. Just do it.
Louis St. Laurent High School is asking, what was the microbacterium that started with a T?
I missed
that. Louis St. Laurent High School is wondering, what was the microbacterium that started with a T?
I had not mentioned microbacterium.
Did you mention any other type of bacteria possibly that started with a T?
No, I'm not sure what our time is here, but what we can do here is the attendees have my email address, and they can email me later on, and I can answer them personally.
Okay, we're not too tight on time, but if there are any questions, then they can ask those, and if not, then people can leave if they need to do so. We originally were going to be tight on time because there's going to be another session, but that other session with another group was canceled. Grey Bruce Health Unit says total coliform. That's what they're getting at with the T. So any last minute questions, or everybody would like to go and have a good day?
Well, thanks for hanging in there. I hope I didn't cover too much. I hope I also made some statements that made you think of what you do and what you should be doing, and maybe rethink of how you do things.
Thank you to all the participants for participating, and thank you to the Royal Bank of Canada Blue Water Project, which has enabled the funding for our webinars. Thank you, Dr. Allen, for presenting such a great and informative presentation. As Dr. Allen said, if anybody thinks of any questions, they can feel free to email him. Also, for those of you who have requested the PowerPoint presentation, and for those of you who wish to email me to do so, Dr. Allen will be taking out any cartoons that are copyright sensitive, emailing it to me, and then I will be emailing it out to those of you who would like the PowerPoint. Thank you all.